All right, so on your sheet, enzymes are specialized macromolecules to speed up chemical reactions in cells. Now, just as important as making them go from small to larger, do we ever want to go from large to small? Is there ever a reason in our body we would want to break something down?
Yeah, so we can use it.
Exactly. When you eat food, you know, if we can't break that down, it's just going to go straight through our body. That's not a usable form. So when we want to break something down, like if we have sucrose, we're always wanting to get glucose. That's a magic thing because we can plug that through our metabolism pathway and get some ATP, which is our energy currency, real fast. So let's say you take in sucrose and you want to break it down. How is that going to happen? Well, this took water out. Guess how you would break down? If you want to break down sugar, what would happen if you put it in a glass of water? It dissolves, doesn't it? And it's going to dissolve into these sugars here. So if you add water, this would actually be plus water. This is the opposite, you're breaking down. So you build up, you take water out. You want to break down, you add water. That reaction is called a hydrolysis. Hydrolysis adds a water molecule breaking the bond. It just it kind of undoes what the other one does. So if I said, when you eat your food, would you think that there are more hydrolysis reactions or more dehydration reactions occurring between the time you put that food in your mouth and digestion and absorption? What do you think? More dehydration reactions or more hydrolysis? Hydrolysis. You know how I remember this personally? When you put food in your mouth, you know how your saliva starts flowing? You know what saliva is? It's a water base. So I just think, right there, that's where it starts. You add that water because you need to chop those bonds to get that in small, usable units. So if I'm ever asking you, suppose I'm going from a protein to amino acids, would it be a dehydration or would it be a hydrolysis? Well, I'll just tell you this. Amino acids are building blocks of proteins. So if I say I'm starting out with a protein and I want to take it down to the amino acid level, what would I do? Hydrolysis or dehydration?
Hydrolysis.
Hydrolysis. Do you understand why? Anytime you go from big to little, it's always going to be hydrolysis. Okay, number 7, polymers are disassembled by hydrolysis. Anytime we say something is metabolized, that means it's chopped into smaller pieces. If we say it's synthesized, that means it's built up. Okay, molecules and polymers can vary depending on what organism you have. So should we stop here, Shanna, and keep going? Okay, we're going to go to carbohydrates now, the next one on your sheet. We're going to talk about each of the 4 under number 1. We're going to talk about each of those 4 different large molecules. The first one is carbohydrates. Carbohydrates, you already know, are sugars. We would call them sugars or starch. The simplest carbohydrates, or the monomer of carbohydrates, would be called monosaccharides. Mono meaning one, saccharide meaning sugar. So the simplest sugars are going to be monosaccharides. If you have 2 monosaccharides joined, what do you think you get then? If you have a monosaccharide like glucose, and you have fructose, which is another monosaccharide, and you join it to get sucrose, this is a monosaccharide, this is a monosaccharide. What do you think sucrose is? If we added more, it would be a poly, but since we've just got 2, let's call it a disaccharide. How's that? A disaccharide? Anytime we just have, it's made up of just 2 monosaccharides, we call it a disaccharide. And disaccharides, when you do this, when you break, you're splitting out this water, right, because we know that. And when that happens, the bond that forms between the glucose and fructose is called a glycosidic bond. Glycosidic. You can call it a glycosidic bond or a glycosidic linkage. The only place that you will see a glycosidic linkage are in carbohydrates. Let me show you. There. If you can see on this, here you have glucose plus glucose. You split out the water, and the bond that forms between them, this is carbon 1. The numbers have to do with the carbon atoms. So always you say this is carbon 1, 2, 3, 4. So this is called a 1,4 because it's 1 and 4th carbon. 1/4 glycosidic linkage. If I ask you what kind of macromolecules or what kind of polymers would have glycosidic linkages, you would tell me carbohydrates. Only carbohydrates have glycosidic linkages. They'll have the same covalent bond, but we call them something different in each of the molecules. This is the one I was telling you about where you have glucose and fructose. And here you have a 1,2 glycosidic linkage. The numbers are not important for you. But just know that the glycosidic linkage is what joins them. Okay, the trademark of the sugar. All sugars have a carbonyl group, number 4, which is, you can write carbonyl, or you can write C double bond O. And they have multiple hydroxyl groups, which that's an OH. So every sugar that you look at, you will see a C double bond O somewhere, and you'll see some OH groups too. Glucose is the most important sugar. Glucose is the most important sugar. Underneath that on your paper, go on and put, this is what glucose is, C6H12O6. And you'll need to know that, because you're going to have to know it before it's over anyway. So just go on and learn it. And it's easy to remember C6H12O6. This says that glucose has how many carbons? 6 carbons, okay? And has 12 hydrogens and 6 oxygens. Now, glucose, let me show you a picture of glucose. This one is not a good one to see. When we go to glucose here, here's glucose right here. We said that it's a sugar and it's got a carbonyl group. Would you classify that as an aldehyde or a ketone? As an aldehyde. So we would call it an aldose if it's an aldehyde. So on number 5B, you would say glucose is the most important, and it is an aldose. So an OSC means sugar. So this would be a sugar that has the aldehyde carbonyl group. A ketose would be a sugar that has the double bond O in the ketone position. You don't have, this is showing you different examples. You can notice here, go on and put on the next one on your sheet, the carbon skeleton can be 3 to 7 carbons long. Glyceraldehyde has 3 carbons, 1, 2, 3. You don't have to memorize these at all, but I just want to show you. Ribose has 5 carbons, which is what's in our RNA and DNA, 5 carbons. Then we have ones that have 6. We also have, these are all aldoses, but we have ketoses that are 3 carbons. It just depends on where the double bond O is. Fructose, the only difference in glucose and fructose is that glucose, the position of the double bond O. Okay, examples of the hexoses on number 7 are glucose and fructose. You want to put those down. And like I said before, disaccharides are joined by glycosidic linkages. They are joined by glycosidic linkages. Usually we draw them as linear, but this is just showing you that some of them are ring forms. If you have a few hundred to a few thousand monosaccharides joined, we call that a polysaccharide. They asked me in the other class, what is lactose, which is the disaccharide, which you all know is the sugar in milk. Lactose, you don't have to write this down, but you may just be interested in this. It is glucose plus galactose. So this is a disaccharide, those are monomers. In lab, if you haven't had lab yet, you will look at yogurt. Yogurt has lactobacillus in it. Lactobacillus are bacteria that have, let me ask you this, if you want to break down lactose to glucose and galactose, what do you think we call the enzyme that you need to make that go fast?
Lactase.
Lactase, okay. So lactase. Now, if you are lactose intolerant, that's because you don't have lactase or you don't have enough lactase. Okay, and you're gonna say, well, I thought you said that it only sped up things, that it can't make something happen that wouldn't happen naturally. So what does it matter if I have lactase? Wouldn't that lactose be broken down eventually? It would, but by that time you'd already be sick at your stomach and not feeling good, and whoever knows what else might happen to you. Yeah, it would happen, but in the body, sometimes we want things done in a timely manner because the middle product might be gonna make us sick or cause us other problems. So if you lack lactase and you eat yogurt, which has lactobacillus in it, and why do you think it's called lactobacillus? It's a bacteria that has lactase. So if you eat yogurt that has lactobacillus in it, the bacteria's lactase will break down the lactose that's in the milk and break it down to glucose and galactose for you. A lot of probiotics have lactobacillus in it, which helps you, just aids in your digestion. So it's important to be able to break down these bonds, all right? So polysaccharides, number 9, are polymers with a few hundred to a few thousand. There are different types of polysaccharides. Polysaccharides can either be for the storage of sugar or for building materials for structures. So if you want, I like to do just like a skeleton outline. So I would do this and I would say polysaccharides. And then you can put, on my sheet, let's see, what did I put? I put A, right, do you see where it says storage? This is the big, underneath them, some of them are used for storage. And that's going to be for storage of sugars or of glucose, whatever. And then if the polysaccharides are not used for storage, they're going to be used something for structural. There are structural polysaccharides, which the main advantage of them is we need them for something for structural purposes. And then underneath that, go on and put, under the storage, put starch and glycogen. The two storage ones are starch and glycogen. Then if you turn the page, you'll see that the structural ones are cellulose and chitin. So first we'll talk about starch, which is a storage polysaccharides of plant. Starch consists of glucose, hooked to glucose, hooked to glucose, hooked to glucose, hooked to glucose. However many. There could be thousands of glucose hooked together. Now, it's more convenient for the plant when it's made glucose, which it's not the first immediate product of photosynthesis, but it will go to glucose. To store it, it's not going to use all the sugar that it makes when it makes its photosynthesis, when it has to produce the sugar. So it's going to stockpile it, and the best way for it to stockpile the glucose is to hook them together and make a large molecule, which we would call starch. Now, when you eat the starch, can you think of a plant that you've heard is a starchy plant? Huh? Potatoes. The reason why is they've got large deposits of starch. Now, when we eat that starch, we've got this bunch of glucose hooked together. Can our body, can we break those bonds and say, hey, we got lots of glucose here that we can plug through our cellular metabolism pathway and get us ATP? Can we do that? Yes, we can. So it's this, they're not eating it for us. Well, maybe they are. We can't prove that they're doing it for us, but they have the starch molecules. You eat it, then your body breaks it down so that it can get the glucose that it needs. Starch represents stored energy. It allows a plant to stockpile the glucose. We do have an enzyme that can break that bond. This is showing you here. Now, we do not store, if we have extra glucose left over, we do not store it as starch, as animals. We store it as glycogen. That's the second one here. So if you want to put that starch is the storage of sugars for plants, glycogen is for animals. If we have leftover carbohydrates that we don't need, we will store it as glycogen, and that will be stored in your liver cells or your muscle cells. Now, if you're on a low carbohydrate or a high or a no carbohydrate diet, the first thing that's going to happen is you're going to deplete your carbohydrate stores, what's stored in your glycogen and liver. How long? What glycogen is stored in your liver and your muscle cells? How long do you think that takes to get rid of what we've stored, our carbohydrates that we've stored as glycogen? About a day. Now, if you'd have asked me, I'd have thought, well, about a month. But that's not true. We're thinking of fat. Yeah, it would take a lot. But if you do not replenish your carbohydrate stores, then after about a day, they're depleted. So we don't have a huge stockpile of that. And people that are for the low-carb diet, they say, exactly, that's what we want. We want you to quit burning carbohydrates. We want you to go and eventually you'll burn some protein. You'll start breaking some protein. And it's harder on your body to do it, but it can use protein and run it through the cycles to get ATP. It can also use fat and run that through. So that's what they're wanting you to do. But then proponents, people that are against it, they say that's too hard on your body and you need a certain amount of carbohydrates because your body has to do the alternate. So, you can talk to one doctor that says it's great and the next doctor says it's not good. So I say it's not good for me because I'm not very happy if I don't have carbohydrates, and it's not good for the people around me if I were to try to go on that. So for me, I believe in a high carbohydrate diet to make Claudia happy. So, of course, I think your mental health has a lot to do with everything, don't you? So that's just my personal take on it. But you do need to know that the glycogen will be depleted in about a day. Now, when we go to the structural polysaccharides, the first thing we'll talk about is cellulose. Cellulose is also a polymer of glucose. I said that starch was a polymer of glucose. And also cellulose. It's a polymer of glucose, which means it's just a bunch of glucose hooked together. But it's different. It's got a different linkage, a different glycosidic linkage than starch does. And if you look here at the slide, this is showing you the starch. This is the only difference. And don't ask me because I don't remember enough about organic chemistry. But for some reason, this bond goes down and this one, the oxygen is up. Now, this is starch. We have an enzyme that facilitates the breaking of that bond. But do you know we do not have an enzyme that breaks that kind of linkage? So when we eat what's starch? Let's talk about starch. First of all, notice here it says it's in the cell wall of plants. So when you're eating plants, you're eating the cell walls too. You can't go through and pick out parts. You eat the whole plant. So you are actually eating cell walls of plant. But you know what happens? We don't get, and if cellulose is a polymer of glucose, we could say, hey, if I eat that cell wall, I should be able to chop up that cellulose and get me some energy, just like I do for starch. Right? It would be a nice idea, but we don't have that enzyme. So we cannot get any, if we eat cellulose, we get no benefit as far as energy component. We do get a benefit because that is it helps to clean out our digestive tract. We call it insoluble fiber. If you look on your food packages and it says how much fiber something has, you know what that's saying? How much cellulose it has. That's what it is. So if you look on your sheet, under 3D, where it says insoluble fiber on food packages is actually cellulose. So a diet in high cellulose is actually healthy for you. Because they say although you don't get benefit from energy, you get that benefit to your digestive tract. Okay, cellulose is in the plant cell wall. It is also the most abundant organic meaning containing carbon, compound on Earth. If you want to jot this down just for your information, plants produce 100 billion tons of cellulose a year. Plants produce 100 billion. I mean, I can't even think of that. I don't understand 1 billion, you know. 1 billion's pretty out there. But 100 billion tons per year is how much is produced. Cellulose is the major constituent of paper. The paper you're writing on is actually mostly cellulose. And cotton is 100% cellulose. Now I said that few organisms possess the enzymes that can digest the bond between the glucose monomers. We cannot. But some prokaryote individuals such as bacteria, and then there are some protists, which are eukaryotic, they can break those bonds. Have you ever thought about a cow? What does a cow's diet consist of?
Grass.
Grass. You know what grass is? There's not a lot, it is not a starchy vegetable. Grass is mostly cellulose. So how does that cow get any ATP from that if it cannot break the bonds? Because cows in their gut have a relationship with certain prokaryotes, or there are some protists that can do it. The cow says, would you like a nice home? If you live inside my gut, then you can have a free home if you'll just chop those bonds when they come through. And they do. So the cow benefits, and also the prokaryotic, the bacteria, or the protist benefits. What about termites? What do they eat?
Wood.
Wood. You know what wood is? Cellulose. Can termites break that bond? No. But you know what? They also either have a certain bacteria that can do it, or they have certain protists that live inside. I mean, can you imagine the thing, where do you live? Oh, I get to live inside of a termite. It's like a trade-off, you know. They break the bonds for them so that they can actually, cows and termites can actually use cellulose to get the glucose out of. Okay, any questions about that? Let's just go on and finish chitin, the last one, and then we'll stop.
What were the three little blanks under cellulose?
It's a major constituent of paper, and it's the only component of cotton. If you read insoluble fiber on a food package, you're actually saying how much cellulose is in there. Now, remember, cellulose is in the cell wall of plants. Chitin is in the cell wall of fungus. It supports them by being in the cell wall. It's a cell wall component. In fungus, chitin is a structural polysaccharide. As structural polysaccharide, it cannot be used as food.
Thank you.
Okay. Chitin is used by spiders and insects to make their exoskeleton. That's kind of like, to me, for that arthropod, I would say he's had an out-of-body experience. Is that not a cool picture? I mean, wouldn't you think that you would like to just say, I'm really tired of this body and I'd like a new one tomorrow? And you just kind of, although it looks pretty much the same size, so I'm not really sure that would help. But their exoskeleton is made of chitin. It's in the cell wall of fungus, like I said. Now, this one is what surprised me. Chitin is used to make flexible surgical thread. Who on earth would have ever thought to say, let's see if we can't use chitin for surgical thread? I'll stop with this. There's no sense in me pretending that I do things right all the time, a lot of the time, so I'll just tell you. You might remember. A few years ago, I was making a multiple choice test out, and the question was, which one of the following is true, I think. And it was, I had some false things, and one thing I put as a choice was, insects use chitin to make surgical thread. Now, I didn't mean to put that. I got too, confused, and I didn't even know it. So everybody's out there serious taking the test, and somebody came up and said, is this true, Ms. Cummings? Because actually that had to be the true one because everything else was false. And I looked at it and I said, well, I didn't tell you that, but the truth is, we don't really know what insects might do we? I can't say that they don't because I've not looked at every insect. But I will say your book never says that, and nobody has ever made that claim. But someday, some entomologist might come up with that idea. And if they do, you just remember Claudia said it happened a long time ago. Now, this is what your homework is. You know chapter 4 forwards and